At QBiC, we take care of your data generation. All we need is your sample and its metadata. The basic workflow in a nutshell. Your sample arrives at one of QBic's partner labs, for example at one of our sequencing or mass spectrometry partners. The sample could be human tissue, a blood or fecal sample, a plant or an environmental sample. Literally any biomaterial. In the wet lab, the experts isolate material from the sample, such as DNA, RNA, or protein. In addition, they perform quality control as an added security measure for you. If the quality is low, we alert you. When the sample passes quality control, the partner facilities process it, generating your dataset. For this process to work as smoothly as it has been designed to, you need to provide two ingredients: the sample and its metadata. Only when we have both ingredients can we start your project. The metadata is crucial, and here's why. Imagine you took a trip through Europe. You photographed everything that struck your interest. Once home, you ask a colleague to create an annotated photo book. They are looking at several photos of pillars. How do they know which pillar was in Rome and which in Athens? The metadata. It tells them when and where the photo was taken and even who took the picture. Your sample is like a folder full of photos, and the data set we generate is like the annotated photo book. We may look at several pillars, for example the genome, proteome, and phenome layers in your sample. To properly analyze, sort, and correlate one snapshot with all the other snapshots, though, we need your metadata. This is the core information that describes your sample, and it includes sample names, for example the label on the sample tube, sample descriptions, the quality measure or measures taken, which type of tissue your sample derives from, and how many replicates are available, which lab protocols or machines were used to prepare it, and many more. Only when we have this metadata can we create a complete photo book for you, with a table of contents and annotations. A complete and multidimensional data set. By providing us with all the metadata on your sample, you enable us and yourselves to find, track, and access your samples at any time, for example for downstream analysis. You enable automation, speeding up data generation and analysis for faster results. With your sample and your metadata, we can give you a multidimensional perspective on your sample. The bigger picture to help you answer your research questions.